Good afternoon, everyone. I'm Ananya. I'm a PhD student in the Chow Lab at the University of Warwick. So today I'm excited to take you on a structural journey of fungal cell wall using solid-state NMR, exploring how the glucans are organized in the cell wall and how they're affected by different mutations. So what comes to your mind when you hear this word fungi? Maybe mushrooms, delicious bakery food items, or something less unpleasant like bread mold, or a disease like athlete's foot disease. But fungi are so much more than that. They are involved in medicines, fermented food and drinks, space, biofuels, agriculture, biotechnology. So their impact vast over science, technology, and even future space missions. Our research interest. So fungi, their cell wall is the structural component and also plays an important role in the pathogenicity and immune responses. And our interest is exploring the fungal role in human health. So we are involved in developing novel applications of solid-state NMR spectroscopies to study the cell wall composition of fungi with relevance to human disease. Did you know that fungal diseases are one of the significant global health issues causing millions of deaths worldwide? In fact, it's about 6.55 million acute cases of several fungal infections annually, out of which 3.75 million deaths due to these infections, which is much more than malaria and tuberculosis combined. And NHS is spending more than 80 million pounds annually in the treatment of these antifungal diseases. The fungal cell wall is not only the structural component of the fungi, but also plays a crucial role in its survival. Unlike bacteria, which is when treated with an antibiotic, the cell dies. But in fungi, if you treat it with antifungal drug targeting one component, which is purple here, the cell will outgrow other component to compensate for that loss and eventually survive. So it is very hard to target antifungal infections. So thus signifying the urgent need to develop better antifungal strategies. The major problem with the development of antifungal drugs is the structural diversity of the fungal cell wall. This figure highlights some of the common pathogens, fungal pathogens, and it shows how each of their cell wall is different. So not many solid-state NMR study is done on the fungal cell walls. These are the three groups that are doing solid-state NMR work on fungal cell wall. It's Tuo Wong group at the US, Mark Balder's group, and Ruth Stark. So their work is majorly focused on studying the fungal cell wall, basically pathogenic, the highlighted one. And we are basically involved in the study of this part, which is the major part and has the potential of broad spectrum, broad spectrum potential. So the key thing is that these are all sugars, and the enzymes making these sugars, they are an ideal target for the development of antifungal drug. Why is that? Because these enzymes are absent in humans. They are critical for cell wall integrity. They are low toxic and has, of course, a broad spectrum potential. For our goal, we have chosen Schizosaccharomyces pombe, which we'll call it as S. pombe. So there are various reasons why we have chosen this, because firstly, it is non-pathogenic. It's easy to handle. Notable model organisms becomes an ideal choice for studying fungal cellular network. It's very easy to grow and genetically manipulate. So becomes a very ideal choice for this purpose. These are the three major components in its cell wall. It's alpha 1,3 glucan, beta 1,3 glucan, and 1,6 beta glucan. And there is no published solid state NMR work on this fungi. So this is what we are trying to achieve. What I do, so I grow my cells on YEA, yeast extract agar plates. expand them in big conical flasks, and then purify or wash in bead beater machine. So these are the samples that I prepare. So these are the two mutations that I induce in the S-POMB. These are alpha 3 deficient mutant and beta 3 deficient mutant, which are respectively lacking the enzymes producing alpha 1,3 glucan and beta 1,3 glucan. We are also interested to see how the wild type S-POMB is affected by different temperatures during its growth. So we are also investigating different temperatures like 24 degrees Celsius and 30 degrees Celsius and trying to see the compositional difference in the cell wall of these samples. So how I prepare my samples? So I use carbon-13 labeling. I use carbon-13 glucose to label my cells. I use 1/10 labeling. The volume I use is 500 ml per sample, which costs about, it's not very cheap, it's 390 pounds per sample. And my samples are basically hydrated, partially hydrated. I study them as it is. And the sample experiment temperature is 15 degrees Celsius to make sure that there is no loss of water during the NMR experiment. So this is unlabeled NMR spectra of the three samples, wild type, mutant, alpha 3 deficient and beta 3 deficient. So we saw that signal to noise is bad on the unlabeled. That is why we wanted to see how labeling is affecting the signal to noise in our sample. So we do see an improvement in the signal to noise. It's much better in the mutants. It's better in the wild type, but it is much better in both the mutants. So thus indicating that, so 1/10 labeling did help a lot. So of course we cannot label 100% because we have to have a balance between sensitivity and resolution. So we chose 1/10 based on the practicality and resolution and sensitivity. So comparing the low and the higher fields, so we do see that when moving on to gigahertz, our signal to noise is much better. And if we look at the carbon-carbon correlation spectra, we do see that the resolution of the cross peaks is much better on the gigahertz. So this shows why we need higher fields for our sample. Further, these are carbon-carbon correlation spectra of the three samples, wild type, mutant, alpha 3 deficient, and beta 3 deficient mutant on the gigahertz spectra. The mixing time is 20 milliseconds over here. And we then try to compare both the mutants to see if the expected changes are observed in the two mutants or not. We do see the changes, like the box, which is showing the beta 1,3 glucan cross peaks. If we look at here, the cross peak intensity is decreasing in the beta 3 deficient mutant, which lacks this enzyme to produce the glucan. And the inverse is seen in the alpha 1,3 glucan cross peak, which is more in the beta 3 deficient mutant and less in the alpha 3 deficient mutant. So the mutations are showing the expected compositional variation in our sample. Further, after looking at the 2D spectra, I went back to assign my peak on the 1D CP spectrum of all the samples. And we observed the peaks of all the three glucans in the sample with different composition, alpha 1,3 glucan, beta 1,3, and beta 1,6. Further, I tend to quantify these glucans in all the samples based on the area under the peak of this spectra. So based on that, we can see that that beta 1,3 glucan is the major component in all the samples, but we do see an effect of temperature change and mutation in my samples. So what we see is that alpha 3 deficient mutant shows very much alpha 1,3 glucan and much more increased amount of beta 1,3. So the cell wall has tried to compensate for the loss of alpha 1,3 by outproducing the beta 1,3 glucan. And the thing is observed in the beta-3 deficient mutant. Further, we also wanted to see how the whole cell looks like. So we grew the same mutation, same temperature changes, and wanted to see the whole compositional change in the whole cell to have an overview of how the cell wall looks like. So in this, we just did not do the purification of the. We did not do the purification. We observed whole cell. Similar labeling, similar sample volume, similar temperature to maintain that consistency. So this is the comparative plot of the cell wall and the cells. So if we look at the cell walls, we just see the sugars. Whereas in the cells, we see proteins and lipids, which is expected, like because we are not doing any washing, we are observing it as it is. So I did the similar compositional analysis on both the cell walls and the whole cells. Let's look at the wild type. So similar observation was seen in the whole cells. But so the whole cells did not show much change in because of the temperature shift, whereas the cell walls did show the difference in the composition because of the temperature. So we are yet to investigate on that by increasing number of batches, by growing more batches and trying to see how it is changing. And then the similar observation was done on the mutants. So we do observe that the mutations behave similarly like the way they are expected to in both the cell walls and the whole cells. We do see some differences in the cell walls and whole cells. So we have different reasons for this. Thing can be that this glucan might be retained in the cell wall. So that is why it's observed less. Further thing is, what else can we find out using solid state NMR? So this is the fungal cell composed of cell wall and cytoplasm. The cell wall is majorly the glucans, the sugars that we are observing so far. So they are tightly packed for using different hydrogen bonding or different interactions. So they are majorly forming the solid part, the rigid portion of the cell. Whereas the cytoplasm is the highly dynamic portion of the cell. If we run solid state NMR on whole cell as it is, we tend to observe both the rigid and the dynamic portion in the cell, which we did. This slide shows a comparative plot of both the rigid and the dynamic portion. So the rigid is the dark blue, the CP experiment, which is based on dipolar coupling in milliseconds. So it highlights the molecular motion in the millisecond scale. And then there is inept spectra, which is observing the J coupling. So it is basically highlighting the molecular motion in the nanosecond scale. And we do see different peaks, different sugars are observed in both the spectra. So the rigid and the dynamic portion are different. The dynamic portion is majorly composed of galactomannan, which has 3 components: beta galactofuranose, 1,6 alpha mannose, and 1,2 alpha mannose. So this is basically involved in responsible for the morphology and the reproductive behavior of the cells. Yeah, so this analysis, this peak assignment has been done on the 2D ineptopsy that I have performed, which I have not shown in this presentation. So the takeaway points of my presentation. So we have seen that cell wall synthesizing enzymes are the prime targets for antifungal drugs, but their compensatory pathways complicate their understanding. And we have also seen that mutations in SPOMB alter the cell wall synthesizing enzymes, and solid state NMR can help us reveal these impact. Solid state NMR can also help us distinguish between the rigid and the dynamic, which is the mobile portion, providing an overall insight of the SPOMB dynamics, which can further help us in designing antifungal drugs that inhibit these specific enzymes. I would like to thank for this opportunity my supervisor, Ying, and our collaborators at the Warwick Medical School, Mohan and Teresa, the amazing solid state NMR group at the University of Warwick. And thank you everyone for listening.